So this is I'm going to try and cover 3 questions here at once. These are questions 2.26 through 2.26, 2.27, and 2.29. So it's kind of a complicated question. So let's take a look here. Consider a diploid cell that contains 3 pairs of chromosomes designated AA, BB, and CC. Each pair contains a maternal and a paternal member. And we're going to use designations like this. Okay, let's see. So we have A from mom and A from dad, or P for paternal, and B for mom and B from dad, C for mom, C from dad. Probably be easier for me to use a D, but I'll use P because that's what the question told us to use. So using these designations, demonstrate your understanding of mitosis and meiosis by drawing chromatid combinations as requested. Be sure to indicate when chromatids are paired as a result of replication and or synapsis. Okay, so in mitosis, this is the 2.26. In mitosis, what chromatid combinations will be present during metaphase? So let's draw a metaphase cell here. And during metaphase, so the chromosomes have already replicated, right, during S phase, which was during interphase. So I can tell I've made my cell too small here, but let's see. Okay, so we're going to have AM right here. You know what? Let's get rid of that. So I'm going to draw the chromosomes right here. So these are the chromosomes that I'm going to map out here, and I'm going to draw a metacentric chromosome for A's, and for B's I'm going to draw a telocentric chromosome, and for C's I'm going to, let's draw acrocentric. Okay, so acrocentric is just the centromere is kind of very close to the end. Telocentric, the centromere is on the end, and metacentric, that's easy. Centromere is essentially in the middle. So these are chromosomes, A from mom, A from dad, B from mom, B from dad, so on and so forth. So during S phase, when the DNA is replicated, then the chromosomes would look like this if they're condensed, right? So during interphase, they're not all condensed, like you wouldn't be able to see them like this. But if they were condensed, right after replication during interphase, the chromosomes are going to look like this, and these are the sister chromatids. And we still count these as one chromosome because there's only one centromere. And it's kind of fun to work with the different colors. So I'll map these out. Okay. Okay, so these are the chromosomes we're going to be working with. Now, this is before replication. This is after replication. So mitosis comes after S phase, right after G2. So we're going to be mapping out these chromosomes right here. The chromosomes in this form. So if this is the metaphase, let's see, this is our cell here. And we want to know what chromatid combinations are present during metaphase. We're going to just map these out like this, or diagram them like this. So there's A from mom, A from dad, B from mom, B from dad. And C from mom, and C from dad. So how many combinations are present? Well, there's only one combination present, right? So here's the metaphase plate, and all the chromosomes are present. There's only one combination. So the next part of that question is, what combinations will be present at each pole at the completion of anaphase? So what's going to happen during anaphase? Well, the centromeres split, right? And then you have, one half of that chromosome, or after they split, we consider them a separate chromosome, going one way, and the other sister chromatid going the other way. And this is for A from mom and A from dad. No, this is A from, sorry, I totally screwed that up. It's very important to know that this is A from mom going this way, and this is A from mom going that way. And this is A from dad. So A from dad is going that way, and the other sister chromatid, A from dad, is going that way too. So we're going to do that for all of them. Centromere splits, A from mom that way, A from mom that way. Centromere splits, sister chromatid from dad. These are B's, aren't they? B from dad that way, B from that dad this way. And you see what's happening here. We are just making, by splitting the centromere and one sister chromatid going one way, the other sister chromatid going the other way, we're making identical cells. And that's the purpose of mitosis. We don't want any changes to occur during meiosis. This is M. So after mitosis, they should be identical. And if we look at these chromosomes, we can see we have an A from mom, A from dad, B from mom, B from dad, C from mom, C from dad. Okay, perfect. So that's one combination. If we look over here, A from mom, A from dad, B from mom, B from dad, C from mom, C from dad. So the same exact combination of chromosomes in each cell, and that's what we want from mitosis. So only one combination is possible. One combination of chromosomes will be present at each pole at the completion of anaphase. And that's going to change for meiosis, as we'll see next. So the next question, 2.27, flows from the last one. So during meiosis 1, assuming no crossing over, what chromatid combinations will be present at the completion of prophase? So draw all possible alignments of chromatids as migration begins during early anaphase. So that's the second part of that question. So okay, let's take a look. No crossing over. What chromatid combinations will be present at the completion of prophase? So in meiosis now, we're not talking about prometaphase. So at the completion of prophase, we're essentially starting metaphase. So this is when the chromosomes are going to be chilling out on the metaphase plate. So let's take a look here. Now take another look at the image or the diagram right there. So that is showing right here, these are the chromosomes during metaphase, and these are them moving during anaphase to eventually end up at the poles of the cell. And this is all mitosis. So during meiosis, well, we're going to be organized a little differently. So during meiosis, and we're talking about meiosis 1 here, so a very important thing happens, right? The homologous chromosomes are paired. So I wonder if I can, I think I can do this. So I'm going to copy these, because we're working with the replicated chromosomes. paste those here. And then I'm going to draw a new cell. Now during meiosis, meiosis has two parts, the reductional phase, which is meiosis 1, and then the equational phase or stage, which is meiosis 2. And because we want to reduce the number of sets of chromosomes in the sperm and the eggs. And so to do that, the cell has evolved a way to align homologous chromosomes and match them up into pairs. So on the metaphase plate during meiosis, we're going to have tetrads made up of the pairs of homologous chromosomes. So here we have A from mom and A from dad. And here we have, let's say, B from mom, B from mom, B from dad, and C from mom and C from dad. And I'll just draw the metaphase plate here. And the microtubules attached here. Well, no, I won't do that. So now this is important. The chromosomes have to be paired up into homologous into pairs. The homologous chromosomes must be paired up, and this is where we have the synapto-nemo complex forming. But at this time, that has already dissolved or been broken down. So the question is now, what chromatid combinations will be present at the completion of prophase? So just one combination is present, right? You have one from mom, one from dad, a B from mom, B from dad, C from mom, C from dad. So only one combination is present at the completion of prophase. So the next question, next part of 2.27 is draw all possible alignments of chromatids as migration begins during anaphase. Now this is where we get a little tricky, right? So let's say this is metaphase, and now the microtubules are attached. And what would happen here? Well, this chromosome would go this way, and say the dad would go this way. So this one from mom would go that way, dad that way. C from mom this way, C from dad that way. So it doesn't have to happen that way, and it's happening this way here because this is the way I've diagrammed the arrangement of the maternal and paternal chromosomes. So what I want to do is show you the different possibilities here. So I will redraw that one I just did for comparison. So we have, if this is the metaphase plate, we have A from mom, A from dad here. Then we have B from mom and B from dad, and then C from mom and C from dad. Okay, so that's one possible arrangement, and then these would be attached to the microtubules, and if anaphase started, well, the chromosomes, moms would all go to the left and dads would all go to the right. So, but we could arrange this a little differently, right? So let's keep A from mom in the same spot here, and well, A from dad will have to be in the same spot. And these should probably be a little closer than I have them diagrammed here. But let's switch the B's. So now we have B from Dad on this side, and B from Mom on this side. And well, let's keep the C's in the same spot. Well, these are acrocentric chromosomes, aren't they? So I will map those there. Okay, so now we have a different arrangement, right? So we have the A and C moms on the left, with the B dad on the left, and A and D from Dad, A and C from Dad on the right, with B from Mom on the right. So we have a different arrangement here. Now there are two more, right? And I won't draw the chromosomes, but I will just draw the labels. So we can have A from Mom on the left and A from Dad on the right. And now we can have B from Dad on the left, B from Mom on the right. And now I'm going to put C from Dad on the left and C from Mom on the right. Okay, so if you look at these carefully, you'll see that we don't have this same combination of chromosomes on the left here in any of the other situations over here. The same thing for the right. So this is a unique arrangement of chromosomes. And we need one more. I'll draw this. I'll have to map this. Oh, let's see. Maybe I can make some more room. Copying this and moving down a little lower here. Okay, I'll slide that over there. Okay, we should have one more combination possible. Can you guys see it? Can I see it? Well, I'll start with putting Mom on the left here again, and Dad on the right. Is there another arrangement? Let's see. Oh yeah, okay. So we'll put B from Mom over here and B from Dad over here. And we haven't done this one yet. So C from Dad over here and C from Mom over here. Okay, so in this fourth one here, we have four possible arrangements now. There's number 1, number 2, number 3, and number 4. So here we got two moms, A and B, with C from Dad, and then we have A and B and C from Dad with A from Mom, and A and C from Mom with B from Dad, and A and B from Mom and C from Mom. And I only mentioned what was on the left because the opposites were on the right, and you can take a look at those for yourself. So there are four possible combinations of these chromosomes on the metaphase plate as migration begins during early anaphase. So four possible arrangements or four possible alignments of chromosomes. So the next part of this question is 2.29. I don't know what 2.28 is. I don't have my book here, but I have these questions written out. I don't know why we skipped 2.28. But 2.29 is to draw all possible combinations of chromatids during the early phases of anaphase in meiosis 2. So that continues directly from 2.27 here, where we've mapped out all the possible alignments here of or orientations of the maternal and paternal chromosomes on the metaphase plate during meiosis 1. So let's say the chromosomes aligned up here, like in situation 1 up here, or arrangement 1, orientation 1. There's so many different ways of words we can use to describe these. And let's go with orientation. So here we have all the M's on one side and all the P's on the other. So all the chromosomes from mom are on one side, and during meiosis, during Anaphase 1, all the moms are going left and all the dads are going right. So I'm going to map that out down here. So as a result, we would have one cell over here, and let's say this is, let's say these are secondary spermatocytes. So and I don't want to draw the chromosomes. Well, I should. I'll draw them. So we have A from mom over here, A from dad over here, and A from mom over here, A from dad over here, and wait a second, I said A, didn't I? B from mom over here, B from dad over there, and then C from mom over here, and C from dad over here. And the reason why this happened is because, you know, this, by random chance, orientation 1 here, this was the way they aligned on the metaphase plate during meiosis 1. So all the mom chromosomes went one way and all the dads went the other. So if these are secondary spermatocytes, so the products of meiosis 1, and then these are the combinations of chromosomes in these secondary spermatocytes. So what would happen next? So the question is asking us to draw all possible combinations of chromatids during the early phases of anaphase in meiosis 2. So these chromosomes, you know, during meiosis 2 are going to move to the metaphase plate. And I'll label these now. So we decided this was A from mom, this was B from mom, and this was C from mom. Now this is how they would align on the metaphase plate during metaphase 2, meiosis 2, and chromosomes would split, and you'd have essentially an A from mom going this way, A from mom going that way, B from mom this way, B from mom this way, C from mom this way, C from mom this way. Now this one over here, you know, it's going to be very similar, right? Except that all these chromosomes came from dad. So this is A from dad, P for paternal, remember that? B from dad, and C from dad. And during anaphase, centromeres would split, and you'd have A from dad going that way, A from dad that way, B from dad that way, so on and so forth. Okay, so we have two right here, possible combinations of chromatids during the early phases of anaphase in meiosis 2, right? So we have one combination here and one combination here. Now we can do the same thing for all of these. So this is going to be one, we already decided this was one combination, this is the other one. And now we could go through that same process and map it out for each of these different orientations of chromosomes during metaphase in meiosis 1, right? They're going to lead to different combinations of chromosomes. Each one of these right here, this would be number 3, this would be number 4, will lead to a different combinations of chromosomes during the early stages of anaphase in meiosis 2. So there's going to be 8 total. So we could draw out all 8 of them if we wanted to, and the diagrams would be very similar. They're just going to have little, the designation, the superscript on all of the chromosomes is going to be a little different in each case. And again, those come from the way the chromosomes are aligned up here in this earlier part of the problem. Okay, so again, for 2.29, that would be 8 of these diagrams. I'm not going to draw out all 8 of them, but you can if you want. Again, just this one right here is this one right here. So you just replicate that for each one of these, and you get the answer.